Bioluminescence and fluorescence are processes involving the biological formation of light. Each process has different means of doing so, and each process has its own potential applications to the medical community. For bioluminescence to occur in an organism, 4 components must be available. The organic substance luciferin is oxidized and catalyzed by the enzyme luciferase to form the compound oxyluciferin and light. Luciferin must be continuously introduced to the organism, either by diet or internal synthesis. An ion such as calcium must be added to activate the fluorescence. This entire light system is called a photoprotein. Bioluminescence is found in organisms in the darkest part of the ocean, such as anglerfish or jellyfish. Bioluminescence allows these deep-sea creatures to communicate with each other or attract prey. Researchers hope to apply bioluminescence to agriculture or even humans. Studying the luciferin fireflies produce, scientists from the Ludwig Institute for Cancer Research inserted the firefly gene that activates bioluminescent light into modified cancer cells. The luciferin causes the cancer cells to emit light, but with an added photosensitizing agent, the combination proves lethal to cancer cells. Though controversial, the effects of gene therapy in this case yield positive results. One could say bioluminescence has a bright future in the medical field. Fluorescence can also be applied to medicine. External UV light is required to excite the electrons in the ground state of an atom. As UV light is absorbed, electrons graduate to an excited state. The Stokes-Shift model displays how when electrons absorb smaller wavelength photons or UV light, they trigger the emission of longer wavelength photons in the form of visible light. When the UV light is present, this reaction allows researchers to detect the applied molecule. For example, in medicine, biomatter in humans can be tagged with a fluorophore or a fluorescent chemical group. The scientists were able to attach a fluorophore to the antibodies in the bloodstream that fight alien pathogens. The fluorophore illuminates the binding site where antibodies attack pathogens through the process of emitting visible light. These findings can greatly advance research in how we treat pathogens within the human body. Even in its infancy, the work in bioluminescence and fluorescence research has limitless potential. Though creatures from the deep sea and glowing insects have adapted and personalized the trait, applying bioluminescence to humans is difficult due to the genetic testing required. Fluorescence, with its more applicable chemical process, would be the better biological light process for medical applications.